Okay, let's see if people will join us now. But okay, let's wait a minute or two to see if somebody joins us. Otherwise, it will be just the small group of important people. Yeah. Okay, here are some people coming. Wonderful. Let's give them another 10, 30 seconds and maybe until 1 minute past 4, and then we start. Yeah. Okay, I think we should start. If people, there's still a few people joining us. Let's see when they will join us just now. Okay, so good afternoon, everyone. Welcome to this webinar of our research group. Today, I'm very happy to introduce you to Chris Rourk. Chris has a exceptional CV. Yeah, because he studied engineering and he did a master's in engineering and then did a doctorate in law. And he's now a patent attorney. But over the years, he still developed a big interest in quantum effects in biology. And today he will share with us some of his expertise on electron transport in certain neuron groups that I can't pronounce. So Chris, we're very happy to have you and very curious to listen to your presentation. Thank you very much.
Well, thank you. I'm Chris Rourk, and I'd first like to thank Dr. Petruccione and Ms. Adams for the opportunity to talk about the hypothesis that ferritin and neuromelanin in catecholaminergic neurons provide an energy transfer mechanism that's used by those neurons. I will pause briefly after I finish discussing each slide, and feel free to ask any questions at that time or wait until the end of the presentation if you'd prefer.
Thank you.
So neurons of the substantia nigra pars compacta, or SNC, produce dopamine, and neurons of the locus ceruleus, or LC, produce norepinephrine. Both neurotransmitters are catecholamines. These groups of neurons are destroyed by Parkinson's disease, or PD, which results in the loss of voluntary movement and cognitive function. There's presently no consensus as to the reason why damage to those neurons has that impact, although it's universally recognized that that is an effect of Parkinson's disease. So in my presentation today, I will first discuss the hypothesis that ferritin and neuromelanin form quasi-ordered arrays in these neuron groups that function like quasi-ordered arrays of quantum dots. Next, I'll focus on the structure of the SNC dopamine neurons, and will show that they have a unique structure and organization that would allow the mechanism to assist with action potential generation in a way that makes sense to the function of that group of neurons. Next, I'll show that the mechanism can release iron in a form that results in an increase in Ca2+ calcium ions that are associated with action potential initiation. Then I'll go over conductive atomic force microscopy, or CAFM test results on SNC tissue that are consistent with the hypothesis. These results don't prove the hypothesis, but if the behavior of the tissue is consistent with the hypothesis, then that's a significant fact. We'll also discuss some current and future work that can help to prove the hypothesis. So the first stop is the evidence that ferritin and neuromelanin could support energy transfer. So ferritin is a very common iron storage protein complex that's formed from a spherical combination of light and heavy chain protein units. Some call these peptide units, but they're larger than what is normally considered to be a peptide. I think a peptide stops at 50 amino acids, and they're about 130, so they're consistent with the size of a protein. Inside of the 12 nanometer diameter protein shell is ferrihydrite, but more detailed view shows there's also other materials that could be characterized as ferrihydrite precursors. the ferrihydrite itself forms 8 structures within the ferritin core that line up with channels that are formed by 3 protein units. So 3 of these 24 protein units interact or align and form these 3 protein channels. But they're not really channels per se, as I'll show you in a moment, but they're points where water-soluble Fe2+ ions can enter. This structure is similar to that of a quantum dot, in that it can be modeled as electrons that can be stored within a Coulomb blockade. And the electrons are prevented from being conducted out of the quantum dot by the Coulomb blockade, and instead have to tunnel or hop. In the SNC, ferritin stores about 1,500 to 1,800 Fe3+ ions. In the liver, for example, it stores 2,400. So the storage characteristics in the SNC are different than in other parts of the body. It can accept proteins in like a p-type doped semiconductor, because the Fe3+ can be reduced to Fe2+ when it accepts an electron. So that's just one of those strange characteristics of ferritin. So now I'm going to switch to a molecular dynamics model that shows an iron atom migrating out through one of the 4 protein channels. So where 4 of the proteins intersect, these channels are formed that allow Fe2+ ion atoms to be migrated out. It's a very complex process. As you can see, it doesn't just, it's not an actual channel that the ions float in and out. And it's driven by an acidic pH. So the threefold channels are driven by basic pH, and they move the iron into the Fe2+ atoms into the interior and store it as Fe3+. And then the release happens when the Fe3+ is reduced to Fe2+, and it's acidic, and it gets transferred out. So getting back to the presentation. The electrical properties of an individual ferritin core result in a nonlinear current response that is flat at low voltage. So this shows these individual cores. They're separated by several hundred nanometers, so they're not really interacting too much with each other. If it's a full of iron, it's called hollow ferritin for some reason, and if it has no iron in it, it's called apoferritin, and the apoferritin curve is flatter. But the important thing is around 0 volts up to about 0.5 volts, there's really no current in response to changes of voltage. So it's flat at that level when you're talking about individual measurements. And this was measured with a CAFM test, where they just settled the probe tip on one and did the current voltage ramp. Now in this configuration, the ferritin was in a compact, somewhat structured or ordered array. You can see these linear structures that were formed. And what happened here was a little different. They used the same CAFM test, but at the certain voltages of the voltage ramp, you had these strange conductivity excursions. There was, I guess, in this paper, not really a clear understanding of why. There was some discussion about quantum effects, but I think, as I'll show you, that there's a good reason for that. This is a very interesting device. It's in a paper by Berra et al., and I'll refer to it as the Berra device. In this configuration, layers of ferritin, which are shown here on the right as 4 layers, and this is an overhead view that shows kind of a jumble of ferritin in each layer, were deposited. And the current was measured from the top to the bottom, as well as from side to side. The side to side is 40 microns, and that's between these 2 electrodes. And top to bottom was just 40 nanometers. So you have about 3,300 ferritin cores side to side, although it's not, as you can see, it's not lined up, but it's a much greater number of ferritin cores side to side than there is top to bottom. The electrical properties, though, are very unusual. Now, top to bottom, there are nanoamperes of current measured, about 0.35 at 3 volts, and it's a relatively linear response. Side to side, there are microamperes, so 3 orders of magnitude greater current measured, and almost 3.5 microamperes at 3 volts, and it's a nonlinear kind of logarithmic curve. So if you consider that the voltage between cores in this configuration, the side to side, is about 1 millivolt, really we shouldn't be seeing anything here. And instead we're seeing much greater current. And this is consistent, as I'll show you, with arrays of quantum dots. And even this really is somewhat consistent, because when you get beyond 4 layers, this drops to 0. So there's no measurable current for 5 layers or thicker. They went up to 9 layers, I think, in the paper. And so for some reason, this stops. They attribute this to tunneling, which I think is probably correct, but they didn't. I've been in communication with them, and I haven't had their comment on whether or not they agree that this is mini banding, which we see in this slide. Now, the drawing on the left here is a 4x4 array of quasi-ordered quantum dots. The size and the spacing is not uniform, but it's not completely disordered. And the computer model conductivity curves are shown here in the middle. This is for an ordered array. So if they were all the same size and spacing, the solid line shows the conductivity that would be calculated. And then the dashed lines are for disorder. And what happens is electron scattering broadens the conductivity curves so that you have some areas that would be 0 in an ordered array that are not 0. It lowers the conductivity, but it's still much greater than 0. Let me make sure I'm covering all the points. The curve on the right here shows that as you pass a threshold voltage, that you have a current voltage response that's logarithmic and relatively uniform regardless of the size of the arrays. The arrays that were modeled in this study were 125 by 25 up to 2744 by 196. So, you know, again, this is consistent. When you increase the voltage on these disordered arrays, you should get, you know, this, you enter into this high conductivity region. It's nonlinear, and it's not directly applicable. This simple model, or even these models, are not directly applicable to the Ferritin measurements that were previously discussed, but they are consistent, I think, with those measurements. So now let's look at SNC neurons. The photo on the left is a cell body, or Soma, of a dopamine neuron. There's a high concentration of neuromelanin organelles, or NMOs, that are distributed within the cell body. The center left photo is a close-up of these NMOs. And they contain roughly 30 nanometer spherical neuromelanin structures. And you can see this is a 1 micron scale bar, so the NMOs themselves can be rather large, you know, up to 1 micron about, but they contain these smaller neuromelanin structures within a bunch of other stuff. The center right photo is taken with electron spectroscopic imaging, and the red is ferritin. And it's actually iron, I think, but it's been discovered by other methods that it's ferritin. And these layers of ferritin that form around the NMOs are similar to the layers in the Barra device. Neuromelanin is a combination of eumelanin and pheomelanin, which form you know, a chain. So it's not just the individual molecules shown there. And both of those are pi-conjugated polymers, which is shown here as an alternating double and single bond. A pi-conjugated polymer has delocalized electrons that are shared across adjacent aligned p orbitals, and they can create a conductor or semiconductor property depending on the specific structure and you know, whatever's mixed in, of course, it shows Fe3 plus is mixed in with neuromelanin here. And that's similar to an N-type doped semiconductor material. The largest soma of the SNC neurons have the most neuromelanin. In this photo of SNC tissue, tyrosine hydroxylase staining shows where there's also a lot of iron, because tyrosine hydroxylase is part of the iron homeostasis mechanism, which controls the level of iron within the cell. Fe2 plus iron is very reactive, and it has to be neutralized so that it doesn't damage other stuff. And the cells have this defense mechanism or homeostasis mechanism. The glial cells between the neurons also contain a lot of ferritin. There's a paper that goes over that that I don't reference here. In my first paper, I assumed a spacing between large soma of 75 microns, which turns out to be very conservative. I didn't know about this additional information when I wrote that paper. The actual spacing is less than 25 microns in many locations. And the hypothesis is that all of these neurons in the soma are coupled by this quasi-ordered ferritin and neuromelanin in each soma and in between in the glial cells. This is just a slice, so there's layers above and below that, you know, have other neurons that would form structures. So that's the hypothesis. These images show the design of a photovoltaic cell that uses quantum dots and pi-conjugated polymers. So here's a pi-conjugated polymer in green and quantum dots in black. And these were designed, of course, to form excitons and to conduct the electrons in one direction and the holes in the other direction to create current, because they're for photovoltaic cells. And they use photons to generate the excitons. Now, an exciton is an electron hole pair that can be formed when an electron gets enough energy to break free of an atom or molecule. And in this regard, quantum dots are considered to be artificial atoms. For an exciton, the Coulombic attraction keeps the electron and the hole together, unless the electron gets enough energy to overcome whatever Coulomb barrier exists and to escape. So the NMOs and ferritin can be modeled as a layer of p-type ferritin that surrounds the n-type neuromelanin. The same process that forms neuromelanin can also create byproducts with triplet state electrons, which are high energy electrons that are prevented from dropping to the next lower energy state because they have the same spin. So that's the dopamine, the melanin, and the iron interact, and it's a very complex process that I could go into if I had more time, but this is similar to what happens in melanocytes that are exposed to UV light, and these triplet state electrons in melanocytes are what causes skin cancer. And in melanocytes, they have to go a much longer distance. They have to travel microns down the dendrites to get to the DNA. In the neuromelanin organelles and the dopamine neurons, they're all in very close proximity, so it's a much stronger effect. I represented the triplet state electrons as excitons as free electrons here. The excitons have a Bohr radius that is directly related to the relative permittivity. And in neural tissue, the relative permittivity is much greater than 1. It can be 80 in the vicinity of the protein, and some measurements show up to 15,000 as a relative permittivity. So whatever it is, it's much greater than 1, and that increased Bohr radius facilitates mini-band formation. So this, again, is a very high-level summary, and there's a lot more detail that goes into this slide, but there's probably several dozen papers that I distilled to get this slide together. So moving on to the hypothesis, in order for the energy transfer mechanism to work, the energy has to move from a higher energy state to a lower energy state. In these neurons, the higher state is the interior of the soma, and the lower state is the voltage common of the extracellular fluid. So neurons are switches, and there's 80 billion neurons, and they all operate as switches in general. They integrate dendritic inputs to reach an action potential, and when they reach an action potential, it triggers a cascade of sodium and potassium channels that propagates in one direction down the axon to the synapses with what's called an efferent cell and the dendrites of that efferent cell, and creates a stimulus that carries that on. Those dendrites integrate the charge, and if they reach enough, then it triggers an action potential in the next neuron. So to understand how the hypothesized mechanism works, let's consider a simple example of 4 neurons with 2 afferent input neurons and 2 efferent action neurons. If all of those neurons are below the action potential, then nothing would happen. They would just sit there. With the hypothesized mechanism, these input neurons can transfer energy so that one neuron could reach action potential and fire, and it would be the one that has the lowest impedance to ground, because that's how electrons flow from high potential to low. And I believe that would be the one with the lowest or the highest level of dendritic stimulation, as I'll show in the next slides. This is a camera lucida drawing of the axonic arbor of a large SNC dopamine neuron. This is the axon's synaptic connections with dendrites. It's a very unusual structure with upwards of 1 million synapses in humans. Because a single neuron typically has around 1,000 synapses, one of these neurons firing is like 1,000 typical neurons simultaneously firing. These synapses have a strong influence on efferent neurons in the neostriatal region of the brain. And that's involved with the control of movement, as I'll show in the next slide. Most of the SNC dopamine neurons don't have this structure and have a smaller arbor with fewer synaptic connections. So this is very unusual and not really seen much. And you can see that the branches form a lot of parallel connections. This slide is a histogram showing the relative percentage of these large neurons. So with the 36,000 cubic micrometer air volume, they make up less than 10%, although as you get older, curiously, about 10% of your large SNC neurons are large. At younger ages, it's about 5%. There's a Parkinson disease bar here, which isn't really relevant. I'm just looking at the healthy normal brain. But the point is that there's just not a whole lot of these. Most of the neurons are smaller, and it's these large neurons that are going to have the lowest impedance. So why is that? Well, the way that we would model the impedance from the soma to the ground would be through the dendrites and the axons. The elevated extracellular potentials will reduce that impedance because they act as a little helping voltage to create, to help the current flow to ground. And so as you have activity around these neurons, it raises the extracellular potential and reduces the apparent impedance seen from the soma. But the dendrites have a much smaller diameter at the root than these large axons. These large axons have a huge diameter, and then they also have these parallel structures that aren't typical in dendrites. The dendrites have more of a distributed tree instead of these branches. So the net effect would be that the axons would have the lowest impedance to ground, and that the activity at the dendrites would lower that even more. So, you know, if the electrons in this mechanism wanted to flow to ground or wanted to flow to one of the soma, it would choose the soma that has the most downstream activity, which again kind of makes sense from a biological standpoint, that you would want to activate the group of neurons that's receiving the most activity to take an action. So let's see. Much of the brain is associated with visual data processing. The signals from the retina are transmitted to the visual cortex, which is the back of the skull, and has a layered architecture that generates neural signals that are associated with shapes, movements, colors, and other aspects of vision processing. So I'll show you in the next slide, these layers create what are called retinotopic mapped neural signals, and it follows the way that the retinal signals are generated with the image data that comes in. And this is a very interesting, processing structure, and I recommend reading up on it if you aren't familiar with it. What happens once you get out of the primary visual cortex, is the retinotopic map signals go in different pathways, such as the dorsal and ventral pathways. The dorsal neurons are structured and connected to detect shapes and categorize them as shapes that have been previously recognized or new shapes. And then the ventral uses neural structures to detect movement, and these paths are in parallel. And all of these parallel paths reconverge at the basal ganglia to help with movement. The substantia nigra is in the basal ganglia. There are other structures, such as the striatum, the globus pallidus, the thalamus. And the cortex is also involved in this pathway, which is the direct pathway. So the direct pathway controls voluntary action, and the SNC is, of course, as you can see, a major component of that. Neural signals of the direct path override the neural signals of the indirect path, which creates neural signals that prevent movement in the absence of direct path signals. So in other words, the indirect path keeps us from having an action unless we want to, unless it's a voluntary chosen action. So the direct path is specific and singular, and the indirect path is non-specific and global. All right, so with that in mind, we get back to the hypothesis that ferritin is able to convert the energy that's transferred into Fe2+ ions, which cause the generation of calcium ions that are associated with action potential generation for an individual neuron. And again, because the ferritin stores iron as Fe3+, when the electrons are transported and localized in one soma, they're localizing in the ferritin, and it reduces the Fe3 to Fe2+, and at some level, the pH increases and it's released. You have to have a substantial number of atoms converted from Fe3+ to Fe2+ for that to happen. So it's not the sort of thing that the Fe2+ leaks out, unless, I guess, if it was a borderline case, but generally you need a lot. And then iron homeostasis neutralizes these reactive Fe2+ ions to create Ca2+ ions. Now, these Ca2+ ions are associated with a special type of action potential generation, which results in a slow recovery of about several hundred milliseconds. The sodium ions create action potentials that have a very fast recovery, and those can fire maybe every several milliseconds. But if the calcium ions activate the action potential, it takes a while longer for the cell to process the calcium ions. So it's a slower action potential. And again, that's observed in the SNC, and also the locus ceruleus, the LC, I haven't really spoken about too much. It's a different analysis, but it's similar to the SNC. So based on all of this, I decided to commission some CAFM tests, which was not cheap, but I thought that there was a good enough reason in the literature to pay some money to have this done. So the tests were performed on fixed SNC tissue samples from AMS Bio, which is in the business of selling tissue samples for scientific research. They were shipped directly to EAG Labs in Sunnyvale, and Dr. Sarah Ostrowski there performed the tests using a Bruker Dimension Icon atomic force microscope. I also asked Dr. Yuming Hua of Bruker, he's an applications scientist, to review the results to confirm that they were not unusual, and he agreed that they appeared to be accurate and not the result of equipment misoperation or contamination. So I never actually touched these samples, and I couldn't monkey with the results. It was all done by others. These are optical images of the tissue that was tested. This one on the left is on glass, and the one on the right is on foil. CAFM testing requires a ground plane, but the glass slides were used to index the location of the SNC neurons. You can see there's a 1 millimeter scale bar here, and these large neurons are 25 to 30 microns. So it was hard to identify them, particularly since I didn't have any staining. I wanted to keep the number of variables to a minimum, so I didn't stain the tissues. We had to use this process to focus in on areas to test. And this is what we got in photographs of the slides on foil. You can kind of see the round structures here that are similar to the large dopamine neurons. The scale bar is 10 microns, and so you're looking at 20 to 30 micron round structures. So these are the large dopamine neurons. We were pretty sure. I wasn't sure if this is a hole or what. I mean, it could be a lot of this neuromelanin pigment. It was on a metal substrate, so we couldn't get backlighting. So for the second test, because of uncertainties, I tested this area, which was dark, noticeably dark in this filtering, but it didn't have these round structures, didn't have as many holes, and then I determined that this was a control sample. Really, it wasn't relevant, and we went back to this for tests 3 and 4. So CAFM testing uses an oscillating probe that's moved across the surface of a specimen in a raster, so it's line after line. The height of the oscillating probe is determined using a reflected laser signal. So the laser signal bounces off this probe, and as it bounces, it can determine what the height is, and you can map the height of the specimen. And that's what it's primarily used for, atomic force microscopy. The conductive atomic force microscopy was from recognition that you could put a voltage on the probe and measure current. And the Bruker device generates 3 current data points per oscillation cycle. One is for the entire oscillation cycle. One is for the period when the probe is contacting the sample. And then one is for the period when there's this peak force. And since there's 512 cycles per line and 512 lines per test, that's 262,000 individual cycles, or 786,000 current measurements. So there's a lot of data here. And to analyze it, they use visualization software. So in the slide on the right, you see height visualization mapping. And you can see that there are these thin areas. And again, it looked like there was something here, but you couldn't tell if it was maybe, something unrelated to the cells or a hole or what. But we're pretty sure this is just the cytoskeleton and other material within the cytosol, which would be removed from the fixing process and the wax impregnation process. So in any event, what's really interesting is the current measurements. And we had these positive current spikes. And you can see this 0 current line here. It's not indicated on the scale bar, but the 0.4 nanoamp current measurement is unusual because the probe had a -10 volt bias. And this is a ground plane underneath the sample. So all of the currents should be negative relative to the node at the probe, with the convention that electrons flow in the direction of negative current. The probe tip was an electron source with a -10 volt bias. And so the negative currents were expected, but these positive spikes were not. And Dr. Ostrowski thought they might be image artifacts, and I wasn't so convinced, so we did the second test. In hindsight, it's clear what was going on. You have these ferritin layers that can conduct tunnel electrons to the probe tip. The voltage, -10 volts across a 5 micron thick sample would be a 2 million volts per meter field, which is high enough to create excitons to liberate the electrons, at least from the pi-conjugated polymers, which have these loosely held electrons to begin with. So the high field strength can give them enough energy to oscillate and form these mini-band structures, and eventually end up in the probe tip, which is going to have a lower impedance to ground than the relatively high impedance of this tissue. So the second sample, we can see that there's none of these thin areas, or there are some areas that are thinner, but it's no neuromelanin we believe is in there, or if it's there, doesn't have enough ferritin around it to conduct. We used a better wax removal protocol because, again, the concern is that several hundred nanometers of wax could interfere with the measurements. But even with a better wax removal protocol, there was no positive current measured. So this turned out to be a very useful control sample that showed that this is not, these measurements were not artifacts of the test specimen or the misoperation of the equipment. At least, it indicates that. This doesn't prove it. We need to do a lot more testing to prove something like that. But it was good enough for me, paying $1,000 a test, to move on back to the areas that had the positive currents. So for the third test, we went back and had the better wax removal, and this time we had much more significant results. And I think the wax removal protocol helped. There were these widespread positive currents. Again, you can't see a zero line in this auto scale, but it's down here somewhere. So you have just an enormous number of points where we're measuring positive currents. And again, I found this very encouraging and decided to do a further test and focus in on one of these areas that had the highest currents. So this was a 78 micron square test sample area. And so I focused in on a 2 micron square for the fourth test, because the resolution here, again, when you're doing 512 measurements across 78 microns, it's not really large enough to detect ferritin, which is 12 nanometers in diameter, or even neuromelanin organelles. Hence with the fourth test. These histograms show the current, and it changes. The peak current is a very short measurement period. The contact average currents are a longer measurement period, and you can see that the current distribution changes. So it's not acting like a linear current where you're measuring the same current across the same, it should have the same shape. So there's something dynamic going on there. The fourth test also yielded very significant results, and this current mapping is similar, lines up with the shapes and sizes of these neuromelanin organelles and the known location of ferritin layers outside of the neuromelanin organelles. This is the height measurement down here, and you can see that we're not measuring surface characteristics, but these are current measurements and not height. So again, this is good evidence. It's not conclusive evidence, but it does show that there's something unusual going on. And then the histogram data for these also shows this dynamic change in current. Now there's more stuff I could do. I found out after these tests that there's a high frequency or a high speed data capture mode that we didn't use. It would be really interesting to see what the high speed, I believe it's like a microsecond sampling frequency, what that shows. But I'm trying to get some additional tests done. There's a university in China that was going to do them, and then this COVID situation put the kibosh on that. So I've got some other tests I'm working on. One is at UT Southwestern, where they can get unfixed neural tissue from a donor who, of course, has passed away and left their brain to science. And the plan is to get a 60 micron thick slice of this tissue and to insert probes into large dopamine neurons, and then use light to generate excitons. So we need a source of excitons to measure any currents. And once you have this unfixed tissue at 60 microns thick, it's too thick to put into a CAFM test. And they don't have one anyway, but they do have these probes, so we might be able to do this test there. And by changing the impedance at these probes, what should happen is all of the current should go down the probe with the lowest impedance, and none should go down the other probes. And this is not what you would expect if it was a classical current. If this impedance was twice that impedance, then the current through here would be half the current through there. And instead, what we're expecting is that there should just be an all or nothing current response. So that was being discussed, and again, the COVID situation came along, and so that's on hold for now. They're not doing anything, they're all working from home. Another thing we could possibly do is create a modified Barra device where instead of one electrode on the right, we have 3 electrodes with insulators between them and the ferritin layers here. And then we would do the same thing. We would change the impedance to ground to each electrode and see if it all goes down to all or nothing kind of current flow. Another thing that should happen is if it's balanced, then no current should flow. The electrons have to tunnel to an optimal path. And if they can't find one, then presumably they won't tunnel to any path. I guess it's possible it could go down all 3 paths equally, but you don't see that in the SNC tissue. So I think it's unlikely that it does that. Again, this will be something that we use to test that hypothesis. Now, Center Habitus elegans is a very interesting animal. It's a 1 millimeter long nematode or worm, and it has been extensively studied because it's translucent and you can see all of its parts, and it's been modeled and mapped and all sorts of interesting tests on it. Now, it has 8 dopamine neurons and 4 are very close to each other. 2 are almost touching, and the other 2 are within several microns. And I don't know what the ferritin distribution is, but this got my attention. So a mutant specimen was created that doesn't create tyrosine hydroxylase. And that was tested to see what would happen because tyrosine hydroxylase is a precursor for dopamine production. And so this doesn't have dopamine. And they found that mutant had jerky movement. It would move too slow, too fast. But when they put dopamine, into the animal by putting it into the environment and it got absorbed, then the movement returned to normal. So the question is, was it the low dopamine or the interrupted electron transport that was causing the irregular movement? And in this regard, the study made this interesting conclusion that the dopamine doesn't signal the speed. It increases the efficiency by which the animals integrate information about their current speed. Well, the dopamine itself can't integrate anything. So it's the dopamine and the neural structures that's doing the integration. And this is consistent with the hypothesis. Doesn't prove the hypothesis, of course, but again, just interesting fact. So if you model these 4 dopamine neurons as a state machine, you can see that there's 16 states as opposed to just 5, which would be if 1 is on and causes movement, and if it's off or they're all off, then there's no movement information coming in. So this would be one way of saying, okay, this is helping to modulate, to integrate this information and to result in a smoother movement. So, again, is this an accurate model? It's hard to say, but you could extend this model to the SNC. And what I've done here is I've taken a picture of an intersection and I've overlaid a very simple retinotopic mapping of quasi neuron cells, 11 by 23 for 253 cells. And when we look at this picture, we know that there's cars and trucks and lights, and you can take a left or you can go straight. You know that these cars have been driving in a certain manner. You know that you have to go somewhere. Maybe your kid's in the back seat yelling. So all of this stuff is going on. And the hypothesis is that all of those neurosignals come in to the SNC and are integrated, and they influence what action is selected. Now, with just 253 neurons, you would have 2 to the 253 states, or about 10 to the 76 states. And that's a few orders of magnitude less than the estimated number of atoms in the known universe. So there's an enormous number of states that can be represented by the SNC receiving all these neural signals if it has this mechanism. The actual SNC has about 30,000 of these large neurons. So 2 to the 30,000 is just an enormous number of states, and it could account for really all of the different experiences, but also all of the different actions. So this mechanism is primarily a voluntary action initiation mechanism. Complex organisms like ourselves have to make choices out of a large number of options. So this is how that choice is made. Our neural structures are, developed over time and tuned by education and experience to make certain choices. and the conscious experience just kind of sits on top of that. And so we think, oh, well, I'm making a conscious choice to turn left here, but actually it's all of our neural structures that are telling us, well, we have to turn left to go, to the store, and this car in front of us is slamming on its brakes, so we should slam on our brakes. So again, this is just how the hypothesis plays out. It's a long way from being demonstrated with actual tests that this is what's going on. And that is the end of the presentation, a little bit early. Dr. Petruccioni, your mute is on, I believe.
Sorry, Chris, thank you. I forgot the microphone. No, thank you very much. It was a fascinating talk. I think that all of us learned a lot. And it was also very interesting to learn about the big efforts you went to proceed with your research. So that was really, really fascinating. Well done. Are there any questions for Chris?
Can I ask a question?
Please.
Have you read anything about the use of quantum, I think there have been some recent papers, the use of quantum dots to treat Parkinson's. Do you think your research has anything it could offer to finding what the mechanism is there?
I haven't read anything about that. Are you aware of some research?
Yeah, I can send you the papers if you want.
That would be very interesting to me.
Yeah, they're using
consistent, I think, with there's another paper that shows that quantum dots help to initiate an action potential. So I'd be very interested to read that.
Yeah, Alzheimer's and Parkinson's.
Yeah, those diseases interfere with the normal operation of the neurons. So giving the neurons additional assistance to reach action potential would help them return to normal operation for a time. Eventually, of course, those diseases kill those neurons, and at that point, they can no longer function.
Okay, well, I'll send you the relevant papers.
Thank you.
Are there any more questions? I was interesting to, sorry, before I ask my questions, I just wanted to tell the participants that are not on the panel and that can't speak by default. If you want to pose your questions, since it's not so many of us, just raise your hand and I can allow you to talk. Just give me a sign and I can let you ask. Otherwise, you can use the question and answer chat facility. No, what I wanted to ask is, our interest is mainly theoretical. Yeah. Do you see? I think there are a lot of places in which maybe we could assist you with some theoretical modeling of these phenomena. Would that be interesting for you?
Oh, absolutely, yes. The mechanism of ferritin within the core has not been modeled. These molecular dynamics models just look at the protein function and they make assumptions about a uniform structure of ferrohydrite, but that would be one area that would be interesting. And we certainly have a lot of test data for the ferritin in various device configurations that could be used to validate any models. But there's certainly a number of other tests that could be conducted.
Mm-hmm. Okay. If you have I don't know, some specific literature of models in mind, if you wouldn't mind sharing that with us, it would make it easier maybe to have a starting point to look into how we could try to model some of these phenomena. That would be very interesting. Yeah.
Absolutely. And also, if anyone else has any questions, tomorrow something occurs to you, feel free to send me an email. I'm glad to respond to questions.
Thank you very much. I know that while you were talking, I was chatting with Yazira, who is one of the attendees of the meeting. And she was very interested in your atomic force microscope imaging, because that's something that that she could do as well in her lab. so that we could offer you a low-cost solution. I don't know if Yazira would like to say something on that. Okay. Yes.
Hi. Okay, perfect. So yeah, a very interesting presentation, and I quite like the measurements you did with the atomic force microscope. The thing is, we have one in the lab at UKZN, and most of the measurements you mentioned, we ideally can perform that as well. So I'm really happy if we could collaborate, and if you have more samples that you would like to test at UKZN, we're happy to actually run them as well.
That would be great. What brand of AFM do you have?
Sorry, now it slips my mind. Yeah, it really slips my mind right now. But like we've been actually managed to go down to like 60 microns, even less than 60 microns on measurements. So we can really go really small samples in terms of depth. So yeah, we'll be really willing to work with you.
Great. The Bruker device puts the voltage on the probe. I've been doing some work with UT Dallas, University of Texas at Dallas, but they have a different atomic force microscope. I'm trying to remember the brand. I'd recognize it if I heard it.
Yeah, is it possible to maybe have a conversation with me offline and we can discuss a little bit more, and then I'll send you the brand that we have as well?
Absolutely.
Okay, perfect.
Thank you, Yazira, for that. Yeah. Are there other immediate questions for Chris? Bethany, do you have more questions? Yes.
Not right now, but I can email as well.
Okay, okay. Thank you very much. I think, Chris, thank you. I think we all need some time to digest the load of information that you share with us this evening. But it was really very interesting, and we will definitely, more of us will stay in touch with you to try to keep our connection going. Yeah, and I see several points.
I'll send a copy of the presentation to you and Bethany, so that you'll have a copy of that to share with others.
Thank you very much. That would be lovely. That would be very nice, because you had lots of references that we could follow up on. Yeah, thank you very much. Okay, Chris. Then I thank you again for your time, for preparing the nice presentation, and I wish you a nice rest of the day in your part of the world, and I wish the South Africans a good evening, because here we are starting. The evening procedures start here just now. Yeah. So Chris, thank you very much again. I will share with you the link to the YouTube video just now, as soon as it is saved and uploaded. Thank you very much.
Thank you for the opportunity, both of you.
Absolute pleasure.
And thank you for all the listeners for putting up with my my presentation.
It was lovely. Thank you very much. Thank you, Chris. Thank you, Bethany, for organizing this. Thank you very much.
Thanks very much.
Yeah. Bye. I then close the meeting now. Thank you very much.